Welcome back to the deep dive. Our mission today is pretty sharp. We're taking a look at some of the most significant medical breakthroughs from this past month, December 2025.
Yeah, and we're really looking for the moments where the scripts got flipped, where the standard clinical thinking was challenged.
Exactly. We've got a story about a therapy that feels potentially curative, another that questions decades of drug safety dogma during pregnancy.
And a really critical warning about one of our most exciting new diagnostic tools. These are big shifts in how we think about what's curable, what's safe, and, what's diagnosable.
Okay, let's unpack this. We're going to start on the cutting edge of oncology. There's this announcement that, frankly, feels revolutionary.
It really does. It's a landmark FDA approval for a CAR T-cell therapy called Breyanzi.
And the efficacy numbers we're seeing here are, well, they're the kind you rarely see, especially in an aggressive disease.
Right. So Breyanzi, the technical name is lisocabtagene maraleucel. It just got the FDA nod for treating relapsed or refractory follicular lymphoma and MZL. This is the really new part, marginal zone lymphoma, MZL.
And for anyone listening, why is that MZL approval such a big deal? What door does that open?
It's a huge deal. MZL is what's called an indolent B-cell non-Hodgkin lymphoma. Indolent just means it's slow growing.
But it's still cancer.
It's still cancer. And when it comes back after the first couple rounds of chemo, the patient's options start to get very limited very quickly. This makes Breyanzi the first and only CAR-T therapy approved for that specific patient group.
So you're going from, continuous treatments that are less and less effective?
To a single infusion, a one-time treatment that could offer a lasting solution.
And the data from the trial, the Transcend MZL trial, it's stunning. They studied 66 MZL patients.
And remember, these aren't newly diagnosed patients. These are people who had already failed 2 or more other therapies, so a highly resistant group.
Okay, so what were the results?
The overall response rate, or ORR, was 95.5%.
Wait, say that again, 95.5?
95.5. I mean, think about that. That means almost every single patient had a meaningful reduction in their cancer.
And for you listening, how does that ORR, the overall response rate, differ from a complete response? Because that's the real goal, right?
Yes, the key distinction, exactly. An overall response is, any response. The complete response, the CR, means no detectable cancer left.
And in this trial?
In this trial, 62.1% of patients achieved a complete response.
So over 6 out of 10 patients, who were essentially running out of options, were left with no detectable cancer after one treatment.
Precisely. And that's what's so exciting, but the durability is what really elevates this.
Right, because a response is great, but you want it to last.
And it does. At the 2-year follow-up, 90.1% of those who initially responded were still in remission. The average duration of response wasn't even reached.
Because so many were still responding.
Yeah, they couldn't calculate an average because the responses just kept going. This is why you're hearing oncologists use the word curative.
Okay, so let's get into the weeds a bit. What makes Breyanzi different? Because not all CAR-Ts are the same.
Right. It's an autologous therapy, so it uses the patient's own T cells, re-engineers them to hunt for cancer cells with a CD19 marker. But the design is unique.
It has to do with the costimulatory domain, right? The 4-1BB.
Exactly. The 4-1BB costimulatory domain. Think of it as like a turbocharger and a bigger gas tank for the T cell. The theory is that this specific piece helps the engineered T cells expand more and persist longer in the body.
So they can keep fighting for longer. And what's the clinical payoff of that design? Better results?
Better durability, definitely. And also, it seems, better safety.
How so?
Well, the big risk with TRT is something called cytokine release syndrome, or CRS.
Yeah.
It's a massive inflammatory storm. The thinking is that the kinetics from this 4-1BD domain leads to a less explosive, more controlled response. And the numbers bear that out. While 76% of patients had some CRS.
The severe grade 3 or higher cases.
We're only in 4.5% of patients. That's a much more manageable safety profile than some of the first-generation CRT products.
But it's not zero risk. It still carries that FDA-boxed warning for CRS and neurotoxicity.
Oh, absolutely. This is still a very serious, specialized treatment that has to be done at certified centers. But for a patient whose other options were palliative, the chance for a single infusion, durable, potentially curative remission, it's life-altering.
It just makes this incredible therapy available to more people who really need it.
That's it.
Okay, let's pivot now. We're going from this super high-tech cell therapy to something that's maybe even trickier, reevaluating decades of dogma around a very common drug.
Yes, specifically for sickle cell disease during pregnancy. We're talking about hydroxyurea.
Hydroxyurea, or HU. This is the standard of care, right? The go-to drug.
It is the absolute bedrock of long-term SCD management. It works by increasing something called fetal hemoglobin, which, basically stops the red blood cells from sickling up and causing blockages. It is very effective.
And yet, for decades, the rule has been crystal clear. If you're pregnant, or even trying to get pregnant, you stop HU. Immediately.
Immediately. No questions asked. And the reason was always about caution.
Why? What was the fear?
Well, HU works by inhibiting DNA synthesis. And way back, animal studies suggested it could be teratogenic, that it might cause birth defects. So because of that theoretical risk, all the guidelines said to stop the drug.
But sickle cell is a brutal disease. Stopping the one drug that's controlling it, that must have consequences for the mother.
Massive consequences. When you stop HU, the protective fetal hemoglobin plummets.
Right.
The disease comes roaring back. Patients get these vaso-occlusive crises, or VOCs.
These are the episodes of just excruciating pain.
Excruciating pain, organ damage. And in a pregnant woman, this causes terrible stress for both her and the fetus.
Yeah.
Hypoxia, inflammation, you name it.
Okay, so here's where it gets really interesting. New findings, just presented at the 2025 American Society of Hematology meeting, are challenging that entire way of thinking.
They are. This study looked at 68 pregnant women with sickle cell. 25 of them had been on HU before they got pregnant.
And the finding was?
They found no statistically significant difference in bad outcomes. Things like preeclampsia or the baby not growing properly. There was no difference between the women who had been exposed to HU and those who hadn't.
So the drug itself didn't seem to be the problem.
It wasn't the driver. Instead, the study pointed to the real culprit.
Which was the disease itself, specifically severe anemia.
That's a core takeaway. It was all about the hemoglobin level. If a woman's hemoglobin dropped below 8 grams per deciliter, that's when things went wrong.
And how wrong?
The patients with that severe anemia had a 47% rate of preeclampsia. That's a dangerously high blood pressure disorder, and a 47% rate of the fetus having restricted growth.
So it sounds like for years we've been focused on a theoretical risk from the drug,
while creating a very real, very immediate risk from the uncontrolled disease. By taking away the mother's stabilizing therapy, you let the anemia get severe, and that's what causes the harm.
The hypoxia, the inflammation from the VOCs, that's probably a much bigger danger to the fetus than the drug itself.
That's exactly the implication.
Yeah.
The authors are suggesting that the most important thing is to keep the mother's hemoglobin stable, which might mean actually staying on the therapy.
But we have to be careful here. This was just one study, right?
Yes.
Small sample size, only 68 patients.
Yes, and that is a critical point. This data is suggestive, it's paradigm shifting, but it's not the final word.
And most of the exposure was just in the first trimester, wasn't it?
Right. Most women stopped the drug once pregnancy was confirmed, so we can't completely rule out some rare later-term effects. But what it does give us is a clear, actionable target.
It shifts the focus from avoid the drug to control the anemia.
Precisely. Stabilize the hemoglobin, or you are inviting a crisis. It's all about that systemic context again.
And our final deep dive today highlights that systemic context in a completely different field, Alzheimer's diagnostics.
Yeah, we've all been so excited about the revolution in blood-based biomarkers, these simple blood tests that can spot proteins like p-tau217 or NFL.
And potentially detect Alzheimer's pathology years before a person has symptoms. It's game-changing stuff.
It is. But a new study, this one in the journal Neurology, is raising a huge red flag, a massive systemic warning.
And the warning is that these revolutionary tests can be deeply flawed if you don't account for one specific organ.
The kidney.
The kidney. So the idea is that chronic kidney disease, CKD, can cause these Alzheimer's markers to build up in the blood.
And lead to a false positive diagnosis for dementia.
Exactly.
So how did they figure this out?
Researchers used data from a big study called the SNSEK study, and they found a really strong inverse correlation between kidney health and the levels of these biomarkers.
And they measure kidney health with eGFR, the estimated glomerular filtration rate. Right, which is just a measure of how well your kidneys are filtering your blood. And the link was clear as day. As eGFR goes down, meaning your kidney function is getting worse, the levels of these neurological biomarkers shoot up.
All of them.
NFL, which is a general marker of neuron damage, spiked the most. But the p-tau proteins, 181 and 217, the ones specifically linked to Alzheimer's plaques and tangles, they were also significantly elevated.
So this creates a trap.
A huge trap. They found patients who were cognitively totally normal, no dementia at all, but who had chronic kidney disease. And their blood tests showed biomarker levels that would absolutely point to an Alzheimer's diagnosis.
Wow. And what's fascinating here is the mechanism behind it. It's so simple when you think about it.
It really is. The kidney is basically the brain's waste disposal system.
Like a garbage disposal.
Ugh.
If the disposal is clogged, the trash backs up, and the metrics look dangerously high, even if the source of the trash isn't the problem.
That is the perfect analogy. These biomarkers p-tau, NFL, they're metabolic waste. A healthy body filters them out through the kidneys. But in CKD, that filtration system is broken.
So the proteins just back up in the bloodstream.
They just accumulate. So a doctor sees a high p-tau reading and thinks, aha, Alzheimer's pathology in the brain, when what they're actually seeing is a failure of excretion by the kidney.
That is genuinely terrifying. I mean, how many people in the age group we screen for Alzheimer's also have some level of kidney impairment?
It's incredibly common. Tens of millions of people, especially older adults. If we ignore kidney function, we risk misdiagnosing thousands of people, causing terrible anxiety, and maybe even putting them on treatments they don't need.
So the study's main takeaway is that an EGFR test, which is a simple creatinine blood test.
It has to become mandatory. It must be a prerequisite for interpreting any Alzheimer's blood panel. You have to know the status of the filtration system before you can interpret the waste.
We should note, though, there was a limitation. The study population was mostly Caucasian.
Right, which could affect how specific correction formulas might work across different populations. But the basic mechanism, renal clearance, that's universal. It just hammers home the need for systemic context.
So what does this all mean? We've covered 3 huge stories today, and the common thread seems so clear.
It's all about context. The most advanced science depends on understanding the whole system.
We saw it with Breyanzi, where understanding the tiny 41BB molecular domain was key to its success and safety.
We saw it in sickle cell disease, where the real danger in pregnancy wasn't the drug, it was the systemic stress of severe anemia.
And then, so powerfully, we saw it with Alzheimer's, where you can't possibly interpret a brain biomarker without first checking the kidneys.
It's never enough to just look at the disease. You have to look at the entire operational system of the patient.
So if we connect this to the bigger picture.
The bigger picture is that this move towards simple blood tests is incredibly powerful, but it's only as good as the body's ability to clear the markers we're measuring. So if a common condition like chronic kidney disease can throw Alzheimer's diagnostics into chaos, it really raises a question for you to think about. What other diseases might we be misdiagnosing right now because we're ignoring the clearance role of the liver or the kidneys? What other critical systemic variables are still missing from our diagnostic panels, causing false alarms or, maybe worse, delaying care for someone who really needs it?